Okay, so the gist of Rocky Mountain spotted fever. Let's talk about the history of the disease. So this is a map of a different color. It's a map of Missoula, Montana. University of Montana is in Missoula. Our collaborator is in this town. There's this river called the Bitterroot River, which goes like this on this old map. And this is called the Bitterroot Valley. Here's what the area looks like. Very pretty. And this is a map of the first study. This is literally a map of the first study on Rocky Mountain spotted fever, which was this disease that the American pioneers sort of like found out about as they were settling the West. So this is in the early 1800s. After the Louisiana Purchase, people are starting to move west and colonize the land. And previously this was occupied by Native Americans. And people have looked and tried to like look into historically, like, did the Native Americans ever sort of like document any incidents of a strange disease or anything like that? Nobody's ever sort of like found any documentation like that. So we don't really know if the Native Americans were potentially just didn't notice it or potentially were immune to it. We don't really know. But it was not documented until settlers started like pushing into this area. Okay. And so what started happening is in early days of Montana becoming a state, a lot of people, Montana had lots of open land, right? That's the kind of like the story of the West, the expansion into the West in the United States history. Lots of vast open lands, and speculators would buy the land, and then it would get sort of like, what would you call, like, they'd buy it when it was sort of like wilderness, and then it would get settled, and then they'd sell it for like high amounts when it was more valuable. And so that was the case in Montana, but there was this issue where there was this river, and on the west side of the river, every one of these black dots was an incidence of a disease that killed a person. Okay, so this was from the earliest study where they're mapping out incidents of this disease. So they noticed that there was this disease that was like killing people in the Bitterroot Valley. It had a high mortality rate, 72%. Okay, that's really high. It's like if you get this disease, you like are pretty much dead. So it was a big issue. And the first study, the first question was, okay, well, let's map out like where these things are occurring. So they map out an epidemiological study showing where the incidences are. And the pattern is what? What's the pattern you see? It's for the most part, like always on one side of the river. It's always on the west side. And this is the east side. So the settlers like realized this. The settlers like realized like if you go to this side of the river and you're like walking around in the area, you like die. So like there's something weird going on here, okay? And that was a big issue, especially for like the economics of the state. And the state cared about this because, right, a lot of this land was owned by speculators, and land prices on this side of the river were very high. It was worth a lot of money if you owned it on this side because it was safe. But if you owned land on this side, your land was essentially worthless because nobody wanted to move there because they would die from this strange disease. So the Montana government wanted to figure out like what was going on, so that, I mean, kind of a greedy justification, but so that they could sort of like figure out the problem, solve the problem, and then the prices of the land could go back up. And then people would want to move west of the river. Okay, so that was sort of like the gist of the early sort of like incidences of this disease and what circumstances funded the earliest studies of this disease. And this epidemiological study is one of the earliest products of the Montana government funding research into this disease. Okay. So when they decided they want to solve this problem, they started hiring researchers. And the first people that they hired, not this guy, the first people that they hired were this duo team named Wilson and Chowing. Okay, and this is just after, this is the early 1800s, so what had just been discovered? I talked about it in this class. What's the context of around this time? What had just been sort of like discovered? Yeah, essentially like vector biology. Like there were major advances in germ theory, and we sort of like understood that like diseases were spread by microorganisms, and there were some specific advances like Patrick Manson in, I don't even know the year now, but the early, I don't know if it's the late 1700s or the early 1800s. I should go back to that lecture. But Patrick Manson had just essentially discovered, okay, filarial worms probably associated with mosquitoes, had suggested that malaria is associated with mosquitoes. Ronald Ross had figured out that malaria is for sure associated with mosquitoes. So those were two like big events in sort of like scientific history. And those two big events started to make people to think like, well, maybe the diseases in my hometown, are actually also caused by things that are spread by arthropods and insects. So that was sort of like the context, the scientific context under which this first group, Wilson and Chowing, go into this region and start studying this disease. And so they immediately noted that this was probably a tick transmitted disease. So Wilson and Chowing were the first people to say, whatever you're seeing here, this is probably a tick-borne disease. They sort of like figured that out. And their hypothesis was that this must be something like malaria. And in that sense, like malaria, in a sense, it would be like some kind of like a protozoan. So they didn't, the other alternatives would be maybe it was a virus, maybe it was a bacteria. And again, like in the early days of germ theory, things were kind of like, well, there's these things called germs. We don't really know what's a virus, what's a bacteria, what's a protozoan. But they kind of had this idea that there were different categories. Protozoans were kind of like the bigger stuff, and then there was kind of like smaller stuff. And they were kind of like, it's probably one of these bigger things. So they started trying to study this, and they developed this hypothesis, but they never proved it. And they wrote a paper suggesting that this is probably a tick-borne disease transmitted by a protozoan. Okay. And this was in the glory days of, again, research was kind of like easy in a sense that you could just go out into like endemic regions where this disease was, collect a bunch of insects. And what their strategy was like, let's collect a bunch of insects, let's just smash them. And they had like rudimentary microscopes, they had rudimentary stains, and they would just stain the smashes and look for like little things that looked like protozoans or like bacteria. Like they would just look for them. And if they saw them, then they'd be like, okay, like maybe this is the thing. Maybe this is the thing. So that was like the style of what people were doing. Okay. And then they sort of, Wilson and Chowing, they stopped studying it, and their research was taken up by this guy named Rickets, this handsome guy. I like Rickets a lot, and you'll see why. He's kind of like a hero of mine. So this guy, Rickets, he's kind of like a rich boy, but he was interested in science, but he also was like kind of like noble in the sense that he decided to go to Montana, and he was like, well, I'm going to spend my time and my money, and I'm going to try to like figure out what is the cause of this disease in this region. So when he first started there, he literally just moved to the region. He didn't have a grant. He just moved to the region and started like doing the research. And eventually he did get funding by the state of Montana. But the good point is he started essentially like by himself, and he essentially got money after. Okay, so he has, I will for sure ask about Rickets on the test because he has a lot of advances in vector biology. And obviously the whole genus of Rickettsia is named after Rickets, this guy. So he has a lot of contributions that he did, and he's worthy of being discussed. So let's talk about some of his scientific contributions. So the first thing that he did, I should zoom out a little bit. The first thing that he did is to study a pathogenic organism. You need a means of culturing the organism. Like, it's really difficult and actually kind of dangerous if your strategy is to just continuously go out into the field and just continuously try to collect things that in theory can kill you with a 72% mortality. So that's actually kind of like a dangerous strategy. So the first step to studying like what this thing is, what this disease is like, let's try to set up like a safe laboratory, a relatively safe, probably would be considered unsafe nowadays, but a relatively safe like laboratory setting through which we can culture the organism that's causing this disease. And the way that they did this is he would essentially go to people who got sick. Okay. And when you got sick, there was a very specific like symptom. Like if this is a human, like what would happen if you got this disease is you would get these spots. That's why it was called Rocky Mountain spotted fever. So he would go to people in the area who got this disease, okay, and he knows the disease is at least like in this person because it's causing the disease. So what he would do is extract blood, and then he would inject that essentially like that mixture, that blood, into guinea pigs, okay? And the guinea pigs would get relatively sick, but they would stay alive. So then once he had sort of extracted the pathogen from the original humans that were sick and injected into the guinea pig, he could then passage this from guinea pig to guinea pig to guinea pig to guinea pig. So he could just inject it into a guinea pig, and then you'd continue the cycle. And then he sort of had like a lab source of the disease for which he could study. So he was sort of like the first person to develop a sort of like a culture system of these rickettsial diseases. And this is a very, very important thing because you cannot study things that you can't culture. Okay. So that's a big point. And there's a funny story with this. Okay. So I'll tell you the funny story, which is when the guinea pigs get sick, they don't die. Like that's another interesting thing is, again, many of these diseases, they're different in different hosts, like they cause different pathologies. And in humans, it'll kill you, but in the guinea pig, they're relatively okay. But they have one symptom, and their symptom is that their scrotums like get swollen. So they get like big scrotums. They get like swollen scrotums. And there was a funny story where I knew this because there's a really good book about this this history called Rocky Mountain Spotted Fever. So I had read this book, so I knew about all this. And one of my friends who studied Ehrlichia and Rickettsia and these diseases, he was defending his thesis, and he brought up guinea pigs or something like that. And then the open question period came. And so to force him to say scrotum in front of everybody, I said, well, what are the symptoms when the guinea pigs get infected with this disease? And then that forced him to say, they get a swollen scrotum. So anyway, yeah, just a funny story. Okay, so now the other thing that people do in the old days when they're studying these diseases is there was a question in kind of like germ theory. There's a question of like, is it a big thing or is it a small thing? And the small things were what we later learned were viruses. And the big things were what we later learned were like protozoans, or like big bacteria. And so one of the very common things to do in the old days was when you had this blood that you could create sickness in guinea pigs or other humans, you could figure out, is it a big thing or a small thing? How would they do that back then? Do you guys have any thoughts? There's a very classical experiment that he did that was very popular back then. Yes. They would filter. Okay, so a filter is just some kind of a mesh. And I mean, I don't know how they manufactured these meshes or whatever they used, but they were able at this time to get sort of filters of different caliber, different quality, different sizes. And so things that were smaller than the filter are able to like pass through, okay? But things that are larger than the filter are not. So the obvious experiment that Rickets did was, okay, is it a big thing or a small thing? So what he would do is he would take the blood from the guinea pig, okay, and he would pass it through a filter, okay, and then you get two portions. You get the portion that's passed through, and then you compare this to a negative control, which is no filter, okay? So you got one syringe with no filtration, another syringe that's passed through a filter, and you inject these into guinea pigs, okay? And what will happen, get a different pen. In this case, the thing that was passed through the filter, there was no disease, whereas the thing that was not passed through the filter got the swollen scrotum and got sick, okay? So he was able to discover that whatever the pathogen is, it's what you'd call, you can filter it out. You can filter it out. That means it's a big thing, or at least it's a bigger thing than the small things that cause diseases, which were viruses. So it's not like yellow fever, not like a virus. This was a big thing, okay? But at that point, okay, there were still, is it like a protozoan, or is it like a bacteria? That was still an open question even after this experiment. Later he would, this is the next slide. It's not, I don't know if it's on any of these slides, but later he would discover that it was a bacteria. So he was the one to clarify what the mistake made by Wilson and Chowing, where Wilson and Chowing said this is probably a protozoan. Rickets was the one to clarify, nope, this is a bacteria. And he would have done that by Germ actually being able to see the Rickettsia bacteria. Okay, so that's another contribution. So one contribution was figured out a way, I should just write these down. He figured out a way to passage in the guinea pigs. So let's just call this passage. Second big contribution was he figured out it's a bacteria. It's not a virus. It's not a protozoan. It's a bacteria. And he did that by filtration and by Germ sustain. Okay, other contributions that he had. He identified the vector as the Rocky Mountain wood tick. So there's a classical experiment in which you can prove this. How would you prove this? This is a classical experiment in vector biology. How would you identify and prove the vector of any disease? How do you take away the vector from an ecological place? That's not possible. You'd have to eradicate the vector. And in which that case, if you knew what the vector was already and you knew to eradicate it, like that's already like getting the cart after the horse.
If you're trying to find what is the vector,
yep.
you'll be looking at the pathogen and see if it transmits from like person from species to species.
Yes, that's one key part. Okay, so you have to somehow demonstrate transmission from host to another host. And you would do that with the vector that you thought was the vector. So what he did is he got guinea pigs, okay? So this is sick guinea pig with a swollen scrotum. And on that guinea pig, he adds some ticks Dermacenter andersoni onto the guinea pig. They feed. You can do them in, I'm trying to think of what the tick life stages, we went over this. Do them early in the larval stage, or you could do a nymph stage. Because remember, all the tick stages feed. So the larvae feed, the nymphs feed, and the adults feed. And in vector biology, the classical pattern is you got to feed twice to transmit a disease. So he would first put some ticks on the guinea pig, Dermacenter andersoni, in either the larval or the nymph stage. And then you would, in the lab, you would let them molt and go to the next stage. So you'd have to have like a way to like culture them. And then you would put them on a new host. So you could put them on a new guinea pig. Okay, so you put the guinea pigs on, or the ticks on a new guinea pig at the second or the next life stage. So if this was the larvae, these would be the nymphs, or if these were the nymphs, these would be the adults. Put them on the guinea pig, and then after they feed, you check to see if the guinea pig gets sick. And if the guinea pig gets sick, then you know Dermacenter is in fact a vector. So he did those experiments. So that's the third big contribution. He figured out that Dermacenter andersoni, the Rocky Mountain wood tick, is the vector of Rocky Mountain spotted fever. Okay, and then people knew, like, okay, the vector is ticks. So then you know, okay, you're not as scared as a land anymore. You just know you have to be very careful of getting ticks off you. Next big contribution. There's another one. Oh, the tick life cycle. He spent a lot of time, he figured out the Dermacenter tick life cycle. So kind of like how Ronald Ross was kind of one of the earliest people to figure out, like, okay, mosquitoes take multiple blood meals. They don't just die after the first blood meal. Rickets figured out like actual like basic life biology, the life cycle of the Dermacenter tick, of the vector. So that's like the fourth big contribution. So he's got 4 big contributions that I specifically point out. Even more than that. Seeing if I missed any points. So interestingly enough, a couple more points about Rickets. This research was essentially like, it went through phases where it was funded or not funded, okay? And after they kind of like figured out the disease, the funding ran dry, and poor Rickets needed something else to study, needed to make new contributions. So he moved to Mexico, and in Mexico, there was a rampant disease called typhus, which was sort of like plaguing Mexico at the time. And we know now that typhus is spread by what we call Rickettsia prowazekii. And its vector, the vector of it is human body lice, okay? But this was not known. And so Rickets actually moved to Mexico to study this disease called typhus. And he figured out that, like Rocky Mountain spotted fever, the disease that causes typhus was also a bacterial disease, and he was the first person to find the pathogen, and so the pathogen was named after him, Rickettsia prowazekii, and that's how he sort of got the genus named after him. It's mostly for his typhus work, but then also for the recognition of the Rickettsia rickettsii. I should write that out. That's an I. Rickettsia rickettsii. That's the species that causes RMSF. And this is the species that causes typhus. They're very similar. They're sister bacteria in the same genus. And they're both transmitted by arthropod vectors. And this was at the time when you studied diseases like typhus, typhus was also like a horrible disease. There were no antibiotics at this time. So typhus had like a very, very high death rate. And imagine studying a disease that there's no cure for, nobody knows what causes it, and there's no treatment for it, and it has a 72% like death rate. So Rickets essentially like signed his own death warrant by deciding to study this disease, and he contracted typhus while he was studying it, and he died from typhus. So he died in Mexico of typhus, and so he was kind of like became like a scientific martyr. And again, another reason why the Rickettsia genus was named after Howard Taylor Ricketts. So I like Ricketts. Good guy. Another point, another interesting point is this research in the Rocky Mountain area, that was continued off and on, and that the area in which they did the research actually became the what we call now the RML, which is the Rocky Mountain Labs. That's a BSL 4 facility for the NIH. And BSL 4 facilities are really rare, so maybe I should just talk about this as well. Different pathogenic organisms are ranked into different sort of like categories, like BSL 1 and BSL 2. BSL 1 is kind of like, we don't really care about it, like yeast, I guess. Maybe it's like BSL 1. It doesn't do anything, we don't care about it. BSL 2 is kind of like, I think actually Zika is a BSL 2. It's kind of like, we should be careful with this, but it's not going to kill people. I mean, there's obviously like clear-cut definitions of these, and as I sort of generalize the specifics, I'm probably doing a disservice. But in general, it's kind of like, okay, things that don't kill you. BSL 3 facilities are things like Orientia, which is scrub typhus, which we're going to talk about. I think Ricketsia rickettsii and Ricketsia prowsecki are also now BSL 3. So these are like dangerous diseases. They will for sure kill you, but we have antibiotics. So like we have ways to treat these now. And then there's BSL 4 facilities, which are like, we have no treatment. It'll kill you. It's very dangerous to work on. This is like Ebola. What else is like a BSL 4 pathogen? You guys know any? What'd you say? I wonder what coronavirus is. I don't.
That's the rats, the one that's in rats, the disease that's in rats.
Oh, bubonic plague.
Plague is not. I think plague would be BSL 3. I think plague would. I think that's Yersinia. We have a lecture on that. I think that would be BSL 3. Most of the BSL 4 things are things we don't have like treatments for. I'm going out of my league here because I don't know anything about BSL 4 facilities. But essentially, the main point that I want to say is that Ricketts's work, Wilson and Chowning's work, and Ricketts's work essentially was continued and led to the formation of the Rocky Mountain Labs, which is a significant research facility in the United States that we have, which is one of our few BSL 4 facilities, which is where we can do research on these really, really pathogenic things. Why would you want it in the Rocky Mountains? The name does sound nice and it's beautiful, but why else would you want it in the Rocky Mountains? It's very isolated. It's very isolated. So when you're studying things like Ebola, you do not want to study Ebola in like a big city. Bad idea. Coronavirus might have come from a BSL 4, but that's a conspiracy theory. We won't talk about that. Okay. Oh, so Ricketts also figured out, interestingly, like whenever you hear like, oh, this is how you properly remove a tick, Ricketts was kind of the one who figured that out, because as he's testing this disease in Dermacentor andersoni, what he notices is that if you pull off a tick and its head is still left in, you can still get the disease even if the body's not there, because the head will continue to feed, and if there's bacteria in that head, it will continue to transmit. So he sort of figured out, okay, when you're going to pull off a tick, you got to insert whatever your tweezers underneath the head and pull it off with the head. That's another interesting thing. Okay, let's talk about this. He also discovered, and if you watched the lecture, I did say this, I didn't mention this briefly. What do we think the mechanism for the rationale of why this disease was on the west side of the river, but not on the right, not on the east side? Did you catch that? Which is? Yeah, well, yes, and yes, and yes. Two things. Ticks, so that would be isolation, and so like geographic isolation. What was the other thing? Yeah, there's actually two bacteria. There's Ricketsia rickettsii, which is the RMSF pathogen. That's on the west side. You find that on the west side. And on the east side is actually a different species, which we call Ricketsia peacockii, not to be confused with Prowsecii. Ricketsia peacockii is non-pathogenic. So on this side of the river, the ticks have Ricketsia peacockii, and on this side of the river, they have Ricketsia rickettsii. And probably the isolation caused by that river is probably the cause of the divergent evolution for that. Because if you look at the tree, they have a common ancestor. Here's peacockii, here's Rickettsii RR. And so they have a common ancestor. And so probably it was a split that caused them to differentiate. And if I'm recalling correctly, the paper that actually sequences their genomes and looks at what's different, if you look at what's different, Ricketsia peacockii has a bunch of pseudogenes. Pseudogenes are what? Genes that were genes, but then broke by maybe like a transposon insertion or maybe like a frame shift mutation or something like that. So it's got some pseudogenized things, and Ricketsia rickettsii has some unique virulence genes. So essentially what I'm saying is that the reason that one would not cause disease and the reason that one would cause disease can be attributed to their genomic content. So there's specific genes that Ricketsia rickettsii carries that causes it to be pathogenic. These are what we call virulence genes. And potentially those virulence genes are either knocked out or pseudogenized or broken in Ricketsia peacockii, or they're completely unique to Ricketsia rickettsii. Okay, so this is sort of theme in bacterial genetics is studying pathogens, comparing them to close relatives that are not pathogenic, finding the differences, and then you can sort of mechanistically figure out, okay, like what are the genes that actually causes this bacteria to become pathogenic? So this is actually something that people have done research on, and it's a hypothesis for why maps like this exist. Okay. Funding ran out, moved to Mexico, and died. So unfortunate. And he died when he was only like 30. So it's like it's a sad story. He had a lot more to contribute. This is the symptoms. So this is what it looks like when you get this. These are the spots. And the reason it causes these spots is because, and this is a pattern you see in these rickettsial-like diseases, these bacterial diseases that are spread by ticks. They get into your bloodstream, and if this is like a vein or like an artery, your veins and your arteries are lined with cells. And what's this layer of cells called? Endothelium. So these are endothelial cells. And Ricketsia and the bacteria that are like them have patterns that they come in, they get in the veins and the arteries, and they infect the endothelial lining of these cells. And they often also infect, like you'll see like Ehrlichia infects neutrophils, which I think are like white blood cells that float around in your blood. And I think Anaplasma infects white blood cells. So they infect things like in the bloodstream and in the endothelial lining. And then once they infect these cells, of course, what happens to these cells? They die, and then some blood spills out, and this would create a spot. So when you see things like this, it's infecting your endothelial lining, blood is seeping out into your skin. That's like what's causing this symptom. And this is another class I've said this before, and this is how they figured out Lyme's disease. This is also a seasonal disease. So doctors will notice oh, people get this in the spring. And that's because that associates with the tick life cycle. So that's a classical case of how you figure out something is a tick-borne disease is all of a sudden people start coming in the clinic during a certain season. They all have this. It's usually because there's a particular arthropod stage at that season that people are getting exposed to. So now we can treat this with antibiotics. So it's not something like we lose sleep over, but we do always worry with antibiotics about like antibiotic resistance. We worry about, you know, there could be a day when we don't have effective antibiotics anymore. And if that's ever the situation, all of a sudden these diseases will shoot back up to 72% mortality again. So these are our serious diseases that we care about. We study them, and they're here in the United States. And we have some incidences of Rocky Mountain spotted fever. Here's Rocky Mountain spotted fever. Here's a map of incidents. Look at Alabama. Yeah, since 2000. We have it in Alabama. I mean, people get this. So it is a serious thing. And if you're not aware of what you're doing and all of a sudden you go out into the field and you get sick and you don't think it's anything special and you don't go to the doctor, you'll end up dead. So it is a serious thing. Okay, I got all that. Okay, let's move on to just quickly some of the other tick-borne diseases. So babesiosis, and this is kind of like that one lecture, which was like the top 10 viruses. I'm just kind of like giving you some other examples of other tick-borne diseases. Babesiosis is like a protozoan parasite. So it's kind of like I think I have it here. It's like the tick-borne malaria. I just want you to know that it exists, what it is. The vector are deer ticks. Deer ticks spread a lot of nasty things. Also, it's spread in agriculture, in livestock with rhipicephalus. Here I'm writing, we don't know the reservoir. Potentially more room for research. Ehrlichiosis. So if you go back to this figure, this is now worth looking at because now you're going to start to see patterns. Okay, so here, this is a tree of bacteria. Tree of bacteria, all in this family, Anaplasmataceae. And I know I talked about this earlier, but now that you've kind of heard of some of these diseases, it's worth like looking at again. Rickettsii. Okay, this is RMSF, Rocky Mountain Spotted Fever. Rickettsia typhi, this is what we call murine typhus. This is vectored by fleas. So this is a different form of typhus vectored by fleas. Orientia, this is what we call scrub typhus, vectored by mites, which the next lecture is about. Ehrlichia, what we're talking about now, Ehrlichia grouping is here. So do you see how these are all closely related bacteria? And what are the common patterns of these bacteria? What are their characteristics? You guys should know that. You guys should for sure know this. What are their characteristics?
Intracellular bacteria.
Thank you. They are intracellular. This is not normal. This is not common to all bacteria.
Obligate.
Yeah, obligate intracellular. That's not a common thing. Most bacteria are not obligate intracellular. This is a unique thing about this family of bacteria. They're all obligate intracellular bacteria. Now they're going to be pathogenic because they have to replicate inside of other host cells. And if they're replicating in your cells, they're going to be pathogenic. Okay. So the one that's not pathogenic, which we also care about for vector biology, is Wolbachia. That's a sister group within this family. And we study this for the sterile insect technique, things we talked about a lot in the past. But one point is that many of the sisters of Wolbachia are these very, very nasty diseases that are spread by ticks, mites, fleas, and lice. So if you study vector biology, you should get very friendly and very familiar with these family of bacteria. The other thing that's worth pointing out about the biology of these bacteria is if you look at this tree, there's this branch which has Rickettsia and Orientia. And these other branches are different, okay? And Ehrlichia, Anaplasma, and Wolbachia, so Ehrlichia, Anaplasma, Wolbachia, live in vacuoles. So in cells, they live inside of a vacuole. Okay. And Orientia and Rickettsia are different. Orientia and Rickettsia live, if this is the cell, they live directly in the cytoplasm. And Rickettsia specifically, there are movies of them. They swim around in the cytoplasm. They use actin filaments. If you've had cell biology, they can like polymerize actin filaments, and they will literally swim inside of cell cytoplasm. And it's actually really cool because what they do is they swim around, and then sometimes they go and they shoot out of cells. And of course, what's next to a cell? Another cell. And so they can swim around and they can explode out of cells and explode into new cells, and swim around and explode out of cells into neighboring cells. So there's actually movies of this. You can see literally movies of this. So it's worth pointing out that Orientia and Rickettsia live directly in the cytoplasm. Ehrlichia, Anaplasma, Wolbachia live in vacuoles. Ehrlichiosis. So the pathogen of ehrlichiosis, the most common species, Ehrlichia chafensis. The vector is amblyoma ticks, Amblyoma americanum. It infects monocytes, which are white blood cells. The reservoir are red-tailed deer. Anaplasmosis, so again, similar bacterial disease. One of the pathogens is Anaplasma phagocytophilum, okay? Reservoir in ruminants, vectors Ixodes, scapularis, and pacificus, so this is the deer tick. And this also infects neutrophils, which are some type of white blood cells. Again, I need to take a class in immunology or something, because apparently there's like many different types of white blood cells. Okay, and this last pathogen, which is worth naming, the common name is Q fever, the pathogen is Coxiella. You hear about this, and if you go to tick meetings, you will see people talking about Coxiella. It's because it's also an intracellular. Now we can do axenic culture with Coxiella, but it's also an intracellular, and it can be spread by ticks. But the more famous way in which Coxiella spreads is it's airborne. It can be inhaled and it can get in the air, and you can actually, like, it can be aerosolized. So it's not most famously sort of like tick-borne, but it also is tick-borne. So it's another thing that you'll see if you go to tick meetings, you'll see stuff on Coxiella. So it's worth knowing like what it is. And it's also an intracellular. So again, things like when people are studying this family of bacteria and they discover new scientific things that are unique to this family, oftentimes they might be some crossover with Coxiella, because Coxiella is also intracellular and also arthropod-borne. So you see these people coming together a lot. Okay, anything else worth saying? Any questions? So that's kind of the finish up of the tick-borne bacterial diseases that are not Lyme's disease. So the next lecture will be on scrub typhus, and the reason scrub typhus is next is because it's within this family, Orientia. And then after the Orientia lecture, we'll lecture about Lyme's disease, which is another bacterial disease spread by ticks, but not within this family. Okay?